All right, folks, so we're heading into some genetics at this point. And when we think about genetics, there's some different types of genetics we can think about, but one of that is primarily the most prominent is what we call Mendelian genetics. And Mendelian genetics is named after Gregor Mendel. Gregor Mendel is what we also consider the founder or the father of genetics. So he lived in the 1800s, obviously, and he was he was not into science. Well, he has a science background, but he was really he was a monk, right? And so as a monk, you spend a lot of time quiet and observing your surroundings. And so he spent a lot of time looking around his gardens and the surroundings at the monastery that he was at. And he started to notice things specifically about different plants. And one thing that he really started to look at was pea plants. So he originally started working with pea plants. So here's just a picture of him with a pea plant. And you can kind of look at some different things. And you can kind of see behind this is what we call a pedigree chart. So it's just kind of an interesting graphic. So when he started looking at pea plants, he started to observe that things were different. So he started to observe that some plants amongst the garden were tall and some were short. So he kind of like started looking at that. He started looking at the fact that some of them had these purpley flowers, and then there were some that had white flowers as well. He noticed that some of the pods were green, but then we had some yellow pods kind of abruptly showing up. He also found that they had some very plump looking pods and some that were kind of shrunken and more kind of shriveled and wrinkled is what he called them. And then when he opened up those pods on the inside, he found that we had peas inside the pod that were nice and smooth, and then some that were wrinkled. And then he found that we also had pea pods that had peas in it that were green versus yellow. So we started looking at all of this variation, and he thought this was very interesting. So what he actually started to do was count. So he was able to kind of put these traits. So now traits are these characteristics that make something unique. And so what he had observed, he already knew that there were tall plants, and then there were short plants, and then there were, different colored flowers. So these are what the traits he referred to them as that were different around just this one species. So he started to actually take some time, and what he did was he had lots of time on his hands. He started counting. He took each trait, so he went with stem length, so how tall versus short they were. And he obviously had a measuring tool to decide what was short and what was considered tall. And he started to actually count them. And when he started to count them, what he decided that basically there was a frequency. That basically he would count out of 1,000 plants, there were 787 what he considered tall, okay? And then he counted 277 short. And so when he did this, that's actually over 1,000. But this is what he counted, right? So he started to count that this was the case. So he saw that the tall had a larger number overall than the short. And so then he kind of went on and he looked at the pod shape. So what did the pod look like? Was it kind of plump and inflated, or was it kind of wrinkled and constricted around the peas? And when he started doing this, he started getting these numbers again. 882 for inflated, 299. So he's getting this really interesting number. Now, he then went and opened each one of those pods, and he counted all of the little tiny peas that were in there. So he called them seeds at the time. So first of all, he counted whether they were round or they were wrinkled. And you can see the number significantly jumps here because obviously each pod has, 5 or 6 peas, possibly, 4 or the variances there. And then he counted of those pods, so he kept track of them, how many were yellow and how many, not pods, peas, how many were green. So he kind of looked at the color. And again, you can see there's bigger numbers. And once he was done with that, he looked at the pod color too. So was it a green pod versus a yellow pod? And again, the numbers were different, okay? And then he looked at the position of the flower. He looked at the color of the flower. So what he started to notice that there was this large ratio. And basically, if you calculated this out, what he ends up finding was that there was always a 3-to-1 ratio. There was 3 times more of the tall plants than they were the recessive plants. And we mathematically could figure that out. And so that's exactly what he did. It's simple math. He took division, accepted the total number, etc., and he figured this out. And so what he started to decide was, when he started to see this frequency, that in every scenario there was a 3-to-1 ratio. This was 3 times more prevalent than this. He started putting names to it. And so the one that was more prevalent, that had the 3 ratio, he referred to it as dominant. And not dominant that means that it was better, but it was more prevalent. It just kind of kept coming out more. There was more frequency of it. And then the other one, which is just a 1 ratio, there was still significant numbers. He called that recessive, that it was just not as commonly frequently coming through. And so when he did this, you could see all of these guys are what we consider dominant and recessive. So he started putting them in a table like this. So the tall was dominant, recessive was short. Tall was inflated, dominant was inflated, recessive constricted. So he started to basically put everything, this is his data collecting from his observation. So he's doing the scientific method, whether he realized it or not in the 1800s. He was observing his surroundings, he observed what was going on, he tested them, and he collected data. He put the data in a table so it's easy to look at and visually appearing. And so he kind of thought there was this idea of blending inheritance, right? Like, so, but what he found that basically, after collecting all that data, is that there was a contrast with this. So he kind of started looking at, different things. So we're talking about generations, right? We have a parental generation, and then a first generation, and then a possibly second generation. And what he started to do, so he started taking those plants that he knew had purple flowers and the ones that he had white flowers, and he tried to cross them. So we tried to breed them and get a new plant. And we have this now, which we call hybrid plants all the time. So he took this plant that had purple, and he knew from his work that purple was dominant. So he gave him these letters of big P, purple, dominant, more commonly prevalent. So he gave him the larger letter. Then he took the white plant, which he knew he calculated to be recessive, and he gave them little lowercase. So a little p is what he had, okay? And when he put these together, what happens is that because of that, he knew that the only thing the purple plant could give for color was purple, and the only thing the white plant could give would be white, right? So he knew that. So when he crossed these two, he had a big P right here, we can see, and a little p right here, because all this plant can give is a little p, and all this plant could give is a big P. And when he did that, he crossed them, and what happened is what we call heterozygous. This is homozygous dominant, this is homozygous recessive, homo meaning the same characteristics. This is heterozygous, meaning that this plant had a big P from the dominant purple and a little p from the recessive white. And so it gets basically one of these little tiny letters, which we will represent as an allele from each parent. But what happened? The plant looked purple. Why did the plant look purple? Because there's more prevalence. There's 3 times to 1 of this. There was a 50-50 chance that this would be purple or this would be white. However, we know that this is recessive. It's uncommon, not uncommon, but not as prevalent. The frequency is not there. And then when we have this particular plant, what is this plant possibly could give its possible offspring? Well, it could be what we call homozygous dominant again. It could be homozygous recessive, or it could possibly give off its own wonderful genotype as a big P and a little p. So this is what we call monohybrid cross, and we'll do another one of these later on as well in the PowerPoint. But basically, here's another example of this. So here's our big P, little p, little p. Well, this can only give one big P, and this can get only a little big P, little p. So we get this heterozygous. Now from there, we do what we cross, and we again get a 3-to-1 ratio. 3 of the plants would be purple no matter what, and only 1 has a chance of being white. So he's continued to see the same type of ratio, 3-to-1, 3-to-1. So let's talk about this. Now, where does our traits come from? Our traits are found on our chromosomes. Humans have how many chromosomes? Or how many pairs of chromosomes? So we have 23 pairs or 46 chromosomes. And different traits are on different chromosomes depending on the trait, and we've learned this from what we call the Human Genome Project, and which is a fairly new project. This is fairly new in the last 30 years or so. But homologous pairs are pairs that are exactly the same. And so in this scenario, we can see here's a parental pair and here's a maternal chromosome. So these are homologous pairs. Let's say this is chromosome, I don't know, let's say it's chromosome 5. Okay, so chromosome 5 from dad and chromosome 5 from mom would come together, right? And we know often we want to basically, so this is homologous, again, meaning the same. And chromosomes usually look like this. They usually look like they're this kind of X shape. Okay, and these are what we call chromosomes, but half of this, right, so if we were to split this in half, these are what we call chromatids. So we basically have 1 and 2 chromatids. Same over here. If that's paternal, this is maternal. If we were to split this in half, this is 1 sister chromatid, here is a second sister chromatid, but that's maternal information. Now, homologous pairs are pairs that are exactly the same. So again, if we were saying this is a particular number, let's say this is chromosome 2, okay? Homologous pairs, I mean, they're exactly the same. This may be chromosome 3 then, and again, exactly the same. The locus, or what we also call the loci, sorry, again, I don't have an actual pen to write with. I'm just using my mouse pad here. The loci, so this should be a C, is a specific location where a trait is found. And so the locations should be the same on both of these traits. So you can see on this, if we're saying this is chromosome 3, this kind of neon green is one trait, the red is another trait, the light blue is another trait. Same thing over here if you're calling this chromosome 2. So one trait inherited. Basically, Mendel, like I said, performed these crossbreeding experiments. Now that he had that original data, he started crossbreeding. He used the true breed, the homologous plants, so that homozygous recessive and homozygous dominant. And there we wanted to what we consider looking at just one trait, because we knew that he had looked at many traits, right? He looked at the color of the peas, the shape of the peas, the shape of the pod, how tall or short the whole entire plant was, where the leaves, where the flowers were located. He just took one thing. And so what he did is he took that one color that he could very well see as the flower, purple versus white. And so when he took that true breed of what he said, homozygous dominant purple, homozygous recessive white, that was that parental generation. And when he crossed them, he had that first offspring generation, which we consider the first phyla, so F1. And then from there, he took those new offspring and created another offspring generation, which would be called the secondary, so F2, phyla second. And he formulated what we call the law of segregation. Segregation means that we can separate things, right? So he took them and he separated them. So he took these traits of the pea plant and he was controlling the two alleles, so the alternate form of the gene. So big P, little p, or maybe he had a big P and a little p together. So like I said earlier, the dominant allele was a capital letter, and that masked anything else. It wasn't better, it wasn't more powerful, it just masked everything else. So it kind of was more prevalent. It basically masked the expression of whatever the recessive allele was, and that what he gave a little p or a lowercase letter. So when we talk about so dominant here, he had given, remember, the big P. This is going to be hard because P, and there's the little p, okay, for recessive. Now, the alleles occur in pairs because we know we have homologous pairs. So, and again, they'll have a specific gene loci. So if they were identical alleles, they were homozygous, like the example he showed us, right? So it was either big P, which was homozygous dominant, or you had little p, which is homozygous recessive. So this was the purple, this was the white. Now heterozygous means that it has two different alleles, and that's when he got to his phyla 1, his first generation. It looked like this. He had that particular plant had one allele that was dominant and the other allele that was recessive. So when we think about that, we have what we call genotype versus phenotype. Now, genotypes are the actual letters, the physical alleles that the actual individual or the trait represents. So that was important, right? The genotype is important. So it was either a big P and a big P. That was the genotype, right? Or little p, genotype. So let me stop here and I will come back on our next video.